Today's topic we will discuss about molecular markers, which is an essential part when it comes to CSIR NET as well as GATE examination. Now, coming to the topic, molecular markers can be characterized into two basic aspects. One is dominant marker, another one is codominant marker. Now, we have to understand molecular markers. They are like our typical RAPD, RFLP, AFLP, etc. There are multiple molecular markers. In today's topic, we will discuss the basic aspect of among these molecular markers which belongs to the dominant type and which belongs to the codominant type, and based upon it we will analyze how the difference in these molecular markers are being established. Now let's dive deep into it. There is a simple process, there is a simple tip that will help you to memorize which among these molecular markers falls into dominant group and which one into the codominant group. Let's find it out. For the dominant markers, there is a simple tip called RASI. Suppose there is a name RASI, shows dominance. Let's deduce from this RASI. R is associated with RAPD, A is associated with AFLP, S SCAR, and I is SR. So these four markers will belong to the dominance segment. When it comes to codominance, we have to find out, there's another tip. Say SSR reveals alleles. From this SSR, S stands for SSR only, which are nothing but mini satellites. Second S will stand for SNP, small nucleotide polymorphisms, and R will stand for RFLP, that means restriction fragment length polymorphism. So in this way you can identify which of these molecular markers belongs to the dominant type and among which belongs to the codominant type. Now, the fact is, dominant and codominant. Now we know how to identify which of the molecular markers belongs to which particular group. From this tip we can state, from the dominant part, we are getting molecular markers like RAPD, AFLP, SCAR, and ISSR. These are the four molecular markers that belongs to the dominant type. And when it comes to the codominant type, we are getting molecular markers like SSR, which is a kind of mini satellite, along with SNP, small nucleotide polymorphisms, as well as RFLP. So these are the classifications. Now, how to identify these molecular markers? Let's say, suppose we are taking RFLP as an example. This is just the introductory class of it. In subsequent classes, we will discuss each of these molecular markers in detail. But for now, let's understand. Suppose RFLP, restriction fragment length polymorphism. These markers, they are present within the genomic segments. These are not the coded regions, they are present within the genomic regions. Suppose capital A, small a. Now, if we identify in this way, suppose capital A, it consists of a genomic segment, and within that region there lies a restriction fragment or restriction cutting site. This restriction cutting site will lead to or will provide us with a fragment. On the other hand, there is another parent, P2. There also lies a restriction cutting site. But there is a difference between these two. There is a difference in between these two cutting sites. So, coming to the fact, when the first filial generation, we are getting capital A and small a. So, both of these restriction fragment length polymorphisms are present within the genomic segments. So, if we run a gel against it, what we are getting? Against the ladder, you are getting a band for P1. Suppose a single band we are obtaining, where there is a single region. On the other hand, P2, it shows band on a different region, as the restriction fragment or restriction cutting site is on a different region within the another parental generation. Now, when these two are met, the first filial generation will give rise to fragments from both P1 as well as P2. So, from this analysis, we are getting that when it comes to codominance, we will receive heterozygosity. We will receive heterozygosity. Therefore, all of these markers will represent heterozygosity. That means both of these fragments, both of these regions will be present, will be visualized within the F1 generation. But when it comes to dominance, we are observing the same scenario. Suppose capital A, small a, there is a region of the gene, and when it comes to another parent, small a, cause the fragments are quite different from each other. And in the first generation, F1, we are getting capital A and small a. Now, the fact is, when it is observed under a gel, what we are visualizing? The molecular markers specifically belonging to these groups, like RAPD or AFLP, whatever it may be, we are observing that from the P1 we are getting a band, from P2 we are also getting a band, but when it comes to F1, we are not observing the recessive entity within it. So therefore, only the dominant parent is showing the band within the first filial generation. This determines the fact that all of these molecular markers will belong to the dominant category, showing only the dominant marker, but not the recessive one. This is how we will analyze the difference in between these two categories of molecular markers.